Okay, class. I'm going to continue on the second segment of chapter 16. We are going to talk about this the structural modeling of DNA, and this is kind of like a scientific inquiry, and I think that you can learn a lot about how the science is being done. Some kind of atmosphere or environment that created in this intellectual society and human interaction, and then beautiful ideas. I think that if you have a time, double helix, okay, by the Jim Watson, okay, wrote it a long time ago. Some people feel that book is not very good, it's offending or something, but I would strongly recommend reading this book if you have a time. So I think that from the discovery of Griffith, and Leon and Avery, and then MacLeod, and also the Hershey-Chase experiment, and they found that DNA is the genetic material. Okay, then how the DNA really looks like and how they structure, then that the chemical structure really account for the inheritance. In other words, how the structure is giving the information to the everything in the cell. So it really comes with we have to talk about these two people, Maurice Wilkins and Rosalind Franklin. Rosalind Franklin is the female physicist at the time working on X-ray, and Maurice Wilkins is also the physical chemist, and they use the this X-ray crystallography and then to study molecular structures. So actually, the Rosie Franklin was the one that produced the picture of DNA molecule using this technique. This is famous. They said photograph 51. Okay, right. So Rosie, they did crystallize the DNA and then take the many pictures, and then this is on like an X-ray film actually. And then they developed it, and then at the time you develop them, then the positive picture is really wet, so they are hanging on the kind of like the thread here, right, the dryer, isn't it? Right. So that's what they used to do, and this is the lady's picture here. Okay, so this is the somehow they took it from, let's say this is the double twist in here, three-dimensional structure. Then let's say this is the round shape, right, overall, right? Then I think that somehow they saw it, from here, okay? So that's how this is a backbone, isn't it? Right, and then you can see something is inside. I mean, this is like $1 million, photograph. And then unfortunately, Jim Watson was stepping by his Maurice Wilkinson's lab. I was told that it was somehow by accident. There are a lot of theories. I think there's the way I understood is that Jim Watson somehow just take a look at it when Franklin is holding this photograph here. So, and then he convinced that, okay, this is the double helix. So Franklin's X-ray crystallographic image of DNA enable Watson to deduce DNA as a helix. Okay, right. And it gives the a lot of picture, and then a lot of image. So this image, made the Watson come up with width of the helix and spinning, and then the location of nitrogenous base, because they are thinking about all this thing, and then when you see somebody shows the sign, then you will know everything, right? So this is the basically that's what they really trigger them to come up with every single idea that they can put it together. Okay, so one of the rules in here, I talked about this single strand I talked about in the previous segment, and then the other segment is going pairing by the Shagaf's rule, but they are anti-parallel. Okay, so they are going on the other strand is going this way, and one of the most I'll use the red. The most controversy or the reason why they were not able to get this kind of structure earlier was that the location of this bases. So it's the nitrogenous base is the one that really makes each nucleotide specific. And but the in the three-dimensional structure, if they aligned it, if they aligned it this way, right? Then what will happen is that the information is going to be buried inside. Think about it. They are going inside, right? So people think that, okay, this somehow what people thought at that time was this base somehow should facing outside, because if they are buried inside, then people think that, oh no, how they're gonna able to access all this information, the buried inside of the double helix. Okay, so it was not really their own idea, but actually people have some kind of information. Actually, at that time, the world most famous chemist was Linus Pauling from United States. He's in Caltech, and they were on this with a lot of money and then a lot of helpers, and then they were trying to get this structure out. And I think that they will still stick on why where is the position of this basis or something. Okay, and I think that they come up with like a triple helix. I heard that back at the time. So anyway, the Watson-Crick built a model of the double helix from this X-ray picture and the chemistry of DNA. It's a controversy, but anyway, the Franklin has concluded that there are two other outer phosphate-based backbone, right? Okay, so this is Rosie Franklin. Okay, the backbone there, and then nitrogenous bases pair in the molecule's interior. That is a really big piece of information. Okay, right. And then I think as Watson, proposed the model that, okay, these two, phosphate backbone is going anti-parallel. Okay, right. So that is the, their, idea. And then I think that base pairing was at first, Watson and Crick thought that A is pairing with A, C is pairing with a C or something. But the such pairing really doesn't result in a uniform width of the this backbone, okay, or base pairing, base stacking. So they come up with a pairing of purine with a pyrimidine, and then forming the uniform, consistent, result in this uniform width consistent with the X-ray data. I think that was the Crick's, idea that, this is going by this. So we have one more model here. Let's say if the purine to purine is too wide, too big. And then if you have a purine to pyrimidine to pyrimidine, it's too narrow. So you're not going to get this. Instead of you are getting this uniform, distance between this, one end to the other end, it goes like this, isn't it? So this is no good. So that's why they came up with the purine goes to pyrimidine and pyrimidine goes to purine. And which purine and pyrimidine pair come from this Chargaff's rule. Chargaff's rule already have the adenine to thymine and then guanine to cytosine. Okay, so this is the what it is, and they came up with this first pair. Okay, and then adenine is binding, interacting with thymine using 2 hydrogen bond. Okay, between these, two possible atoms, and then this guanine to cytosine is the stabilized by 3 hydrogen bonding. So this is a little bit stronger than the others. Okay, so this is a little bit stronger, weaker, bonding here. Let me continue in next segment. Thank you.